Great. All right. Well, Dr. Squires, thanks for joining us to give a recap of the scientific meeting that we had in April. It was a big success, and I look forward to the next one. But you brought up a good point. This is a lot of information that was thrown our way, and some of it was rather technical, which is exciting. But also at the same time, we're listening to it, kind of nodding our heads, saying and then we might not really understand what it means and what that means for us. So I appreciate you taking the time to kind of give us a recap and then answer any questions we may have.
Absolutely. So again, it's always a pleasure to join the network. I don't know if those heard me discuss with Emily. I wanted to be informal about this. I didn't necessarily want to make a formal presentation to give another talk to you guys, right? You just sat through a whole bunch, and didn't want to necessarily kind of re-give the talks that were given. I think they were all excellent, and I think presented a lot of really exciting and fascinating work that's being done in the field of PFIC research. But I also recognize that some of the technical aspects of it may have been at the time, either because we ran out of time to ask questions, or because maybe it wasn't quite understood at the time, and only later, after you had a chance to kind of decompress a little bit, did you kind of think, well, that was interesting, but I didn't get a chance to follow up with that person about their talk. So wanted to just kind of be available and make myself available to you guys if there was any thing I could do to kind of help further along the discussion of really what we're doing, and make sure that you guys got the most out of the meeting, and we're understanding of the efforts that are underway to hopefully change the lives of kids with PFIC. You're still muted, so not only do you have to also unmute, there's a couple.
Thank you very much. For those who missed that, I started the meeting without the headphones in my ear, which made it very challenging to hear. Thank you for bringing that back up. I think the first, let's just go through the agenda, and maybe I'll do kind of like a Q&A through some of the talks, and then we'll open it up. I think one of the big questions, overviews that I think would be helpful to understand is, who is the Children Network and who is the Napa Consortium, and what are they doing for us?
Yeah. So, the Children Network, children stands for, so the Children Network, children stands for the Childhood Liver Disease Research Network. So when you see it written, right, that's why some of it is capitalized and some are lowercase. The Children Network is a multi-center NIH-funded consortium. And so because it's NIH, it's mostly just North American centers, although Sick Kids in Toronto is also included. And it includes about 14 centers. And the history of children, and it predates me, right? So I think someone like Ben Schneider or Ron Sokol would even be able to give more insight. But children started out studying another rare liver disease in children called biliary atresia. And at the time, it actually had a different name. It was called BARC, which was Biliary Atresia Research Network. And over time, what it has become is a research network whose focus is really rare cholestatic liver disease in children. So while biliary atresia is also still studied within the network, it's expanded now to include diseases such as Alagille's, diseases such as the PFIC disorders, diseases such as alpha-1 antitrypsin deficiency. And these are all liver-based diseases which cause children to be jaundiced. This is also where some of the work of the newer PFICs, right, TJP2, USP53, MYO5B, this is where this work has really been pioneered, because we also enroll children who have cholestasis and who maybe itch, but who don't have a diagnosis yet. And we haven't identified that genetic or gene mutation that's causing the abnormality. And so there, we recognize that, we don't know everything yet, as we often talk about. And so having the ability to go back and say, here's a child whose phenotype fits very much in the vein of PFIC, But when we look at the kind of, again, the three historical, FIC1, BSEP, and MDR3, they didn't have any of these. And so through some of these advanced genetic techniques, I have been able to identify some of these other genes that are affecting children and causing this disorder. So again, children is an NIH-funded study, and so because of that, there's some rules and restrictions and things that need to go into whether or not a child is enrolled in that study. You have to consent them. It is a prospective study, which means we see you, we consent you, and then we follow you going forward. Oftentimes, we are collecting clinical data for the study, right? So there's additional data that we might not collect clinically that we say, hey, you know what? We want to look at bile acids in these kids. So we start collecting bile acids, or we will look at specific markers moving forward. This has also been a network just because of its productivity and because of the groups that are involved, that there have been some partnerships with the pharmaceutical companies. So again, some of these drug companies testing the IBAT inhibitors have looked to partner with the Children Network because we have this group of children with these disorders, and the infrastructure is there to do some of these rather complicated, to be honest, studies. So that's kind of children. NAPID is a little bit of a, it's a different structure. So what NAPID is a retrospective study. So if anybody's also familiar with the GALA study in allegeals, I think this is another example of how these registry studies work. So in NAPID, for example, you aren't consented. They don't identify you and then enroll you and then collect information prospectively. What they do is they go around to all the different sites and they say, and NAPID is mostly in Europe. It's mostly a European-led study, but we are obviously collaborating with them in the Children Network. But it's where they kind of go to a center in Germany, a center in Switzerland, a center in Italy, and they say, hey, how many PFIC patients do you follow? And can you go back in their charts and pull out a bunch of clinical data that we can use to kind of apply statistical things to try to understand. So there's advantages and disadvantages to both types of studies. Something like NAPID, that's why if you read a NAPID paper, their numbers are going to be really, really big, right? And so we'll think, oh my gosh, all these patients, are enrolled in this study, that we're going to have all this information. Not really, right? Because it's actually more of a fractured way of doing things, because you have to kind of just go back in time and figure out what's been done clinically, and then kind of apply that to whatever question you're trying to ask. Versus in something like children, you can say, this is a question I want to ask, let's follow forward and see how that question is answered. But obviously a lot of really good information can come out of both types of studies. And although there may be similar overlap type of work, I think that depending on the kind of question you want to ask, either NAPID or children may be better situated to answer that question, but both are still, I think, generating really good data about these disorders, obviously. Got to hit the mute.
This is not my day. Sorry about that. Do you happen to have an example of what you've learned from the data you've collected from children, or from what NAPID has learned from the data that they have collected?
Yeah, I mean, again, I think I'd point to several publications that they presented at the meeting, right? I mean, I think so. I think both children and NAPID have looked at kind of natural history studies, right, to kind of see what is the natural history of these disorders. Here, to be honest, NAPID may be a little bit. The numbers are going to be bigger in NAPID always, right, which is challenging. But I think children, if you want to think about it from a purely scientific standpoint, is maybe a little bit cleaner. The data kind of gets scrubbed. The data is inputted in a way that there's a lot of assurances that the data is real, right? That the numbers that we see are in fact the numbers. Sometimes when you go back and you do retrospective data, for example, and again, if you're looking just at how the bile acids look after a diversion procedure, right? In something like a NAPID trial, you're really dependent on when those bile acids were checked, and they were checked for a clinical reason, right? So I'm like in a chart 3 years ago after a kid had a diversion, and I see their bile acids were, elevated at this time point. Well, they could have been elevated because the kid had a cold, had something else going on, and there was a confounder in that. But the data set probably doesn't get granular enough to understand that. It'll just say, at X time point after their diversion, the bile acids were this. And so you have to be a little bit careful when you do this retrospective data, because you don't really know why these labs were drawn, or you may have multiple blood draws over a time period, right? So if the patient's in the hospital, right, or the patient, had some sort of clinical event, I may check labs every other week for a period of time. And often you don't input all of those numbers into the data set. You kind of pick one because they want to have numbers that are like, at a year after diversion, at 2 years, 5 years, and 10 years. And that's how they build these kind of curves and these longitudinal pictures. So, whereas in children, the time points are much more defined and we see them solely for clinical purposes. But both are probably pretty good at natural history studies. And I think there can be information learned from both of them about the natural history of these disorders. What I think children is much more adept at being able to do is looking at new genes, right? And the reason I say that is because, again, we have blood samples and very detailed phenotypes or the physical manifestations of these disorders that we keep and store for later, so that for example, when the first reports of TJP2 came out, we went back and all of the children in the children network who had samples stored that had cholestasis with the low GGT and said, hey, we should look at TJP2. And we looked very carefully at TJP2, and lo and behold, we found a few kids, right? As USP53 has kind of emerged as another phenotype, we've gone back, and I think Laura Bull just published the children report of, it's only a handful. These are still very small numbers, but I think a half dozen or so USP53 kids within the children network that weren't previously identified to have that disorder. So I think that, this is another area where, although, the early reports are, for example, the FXR, or even the USP53, you have like 1 or 2 patients, right? Or one family. When you have something like the children network that's collecting samples, you can kind of go back and look more broadly at multiple patients across multiple time points. And then also kind of, look at their clinical data that's been very well inputted to kind of see how that evolves over time. So advantages of both, and I think, depending on some of the questions you want to ask, would approach the different networks, to answer whatever question you want.
Okay. Thanks. And so I think you started talking about the different subtypes, the different genes. And Dr. Thompson started out this series with the spectrum of the genetic cholestasis syndromes, the origin of PFIC, and it was entitled PFIC. And so I'm wondering, maybe it's a good segue into talking through that, given a quick breakdown as to that spectrum quickly.
Yeah, I mean, again, I don't think this group needs a lecture on the spectrum of this disorder. You guys live with it every day. There you go. Sorry, hold on. Sorry, in a second, I must need to. Can I do one quick phone call? I'm sorry, I just got. I apologize.
No problem. Dr. Squires is actively on service right now, so he has patients that he needs to respond to, which is what we're letting him do. So let's take a moment.
Sorry about that. I'm back. Yeah, so I mean, I think that the spectrum of PFICs is something that I don't think that you guys need too much education on. You guys know the wide variation. But I think we still are learning all the time about some of the newer contributions of these defects to disease, right? So whether it's a new gene, that I know is kind of touched upon it as we've understood the new genetic contributions, or the kind of emerging contributions of these defects in something like adult populations. And I think there's been some papers about that. And so this kind of textbook classic example of someone with PFIC who has very progressive disease that presents in infancy and requires liver transplant in the first or second decade of life. And I'm looking at the PFIC1 and the biceps that were classically described. That still exists clearly. But there, we now fully understand that there are patients who don't fit that mold, who still have defects in these genes. And this is the challenge of genetics, in that there are different types of mutations, right? This is where we use words like truncating, right, or missense. These are kind of other technical terms that describe to a geneticist how the change in the DNA, how the change in the blueprint of the gene affects the protein that's being made, right? And so if you have a protein that is interrupted, so it's not made at all, you have absent protein. It's not there. That person is going to be much more affected potentially than someone who has a change that causes the protein to misfold, for example, right? There they make the protein, it's just made a little wonky. So it may retain some function, but not perfect function. And so this is why when you get these genetic reports back and it says, yes, there's a defect in the BCEP gene. Well, okay, you need to dig a little bit deeper though and say, what is that defect? How is it affecting the protein? Because that's going to impact potentially the phenotype and the prognosis for this patient. And as we're learning again, treatment options, right? Whether something like diversion it may or may not be helpful, it depends somewhat on we're learning the genetic defect that's causing the disease in the person.
And that ties back into what was presented. I think Dr. Burkad presented it with his Napa data. How what they're learning, you're learning from this natural history data and from following patients who have taken medication or had surgical intervention. In the last 3 years, it seems like there's been a lot of advancement in understanding which interventions could help for which genotypes, right? But only in certain subpopulations. There's a lot that's understood about BCEP now in terms of BCEP 1, 2, and 3, but there's still a ways to go to get there for the other genes. Is that correct?
So exactly right. So I think the broader terminology that we're talking about here that you'll hear us use a lot is something called the genotype phenotype correlation, right? So how does the specific gene defect correlate with the physical manifestations? And those can be just the degree of jaundice, those can be the clinical progress, those can be response to treatment interventions, whether surgery or medical. So that understanding of genotype phenotype is not unique to PFIC. We are always in any genetic-based disorder. That's something that we look to really try to understand more, and something that I think the Napa group is really expanding our horizons on as it relates to the data that they're learning. And you mentioned the BCEP, 1, 2, and 3. Just to be clear, that those are just made up terms, right? To describe patients with specific defects, right? BCEP 1 is a specific genetic defect. BCEP 2 is a specific genetic defect. But it's easier to say BCEP 1 than U.P50, like all the different dots and letters and numbers that describe the defect in these children. So I think this all comes down to, again, recognizing that when we call something at its most broad terms PFIC, or when we call something BCEP, or when we call something Byler's or FIC1, these are all buckets, and within the bucket there is likely variability that should be accounted for, or that we're trying to understand more.
Well, yeah, it's some of the most relevant work that applies to our lives as patients. I think my story is pretty well known now, but it's changing a little bit in terms of what we know or what's known now versus what was known when my daughter was diagnosed 10 years ago. So I see this data, and the way that I take that in as a parent is that if we would have understood, or if you would have, we're all one community. If it would have been understood, this level of, this intervention may or may not have worked for this kind of subgroup of PFIC, it would have changed my daughter's journey, right? There probably would have been certain interventions that would not have been done or would have been done because of the information that you have now on BSEP. And so I think it is some of the most relevant, research that's been published to us as a patient population in terms of understanding how this is going to affect our lives going forward, if that makes sense. I think that was brought up quite a bit throughout the roundtable discussions, is the uncertainty in the pathway, right? It's hard for us to know what to expect. And data like this brings hope to that, allowing us to be able to make more educated decisions together. So yeah, I think that's a pretty good overview of that beginning introduction. I think if we move into kind of the first block of talks, we had a lot of pretty technical talks here. Maybe just I'll take this in for a moment and kind of just allow you to maybe summarize some of these talks.
Yeah, I mean, so I think, Dr. Yin in the Cincinnati group, right? I mean, I think this is something that they've been trying to do for some time, which is come up with these pipelines. And so, here, the question is, you have a child who is cholestatic, and you send off all of the other, PFIC genes, and nothing comes back, right? No hit. Doesn't look like they have PFIC. Or doesn't look like they have any of the known genes that we associate with PFIC. So then, so generally, like, how we work these kids up, right, is have a kid come in, they're jaundice. Likely at some point they have one of these targeted gene panels, right? And so this is kind of the first step in genetic testing. And what these panels do is they generally test for a collection of genes that associate with the phenotype. So they have, targeted gene panels for seizures. They have targeted gene panels for hemophilia. They'll have targeted gene panels for, pick your disorder. There's a targeted gene panel for cholestasis. And so this tests for currently about 70 different genes. And many of them, they don't have off the bat, right? Because there's, different phenotypes. Kids who have PFIC don't look like kids who have, primary, ichthyosis, right? Or some of these other rare disorders. But it's just kind of cheaper and easier to send off for all these genes at once. So you get the gene panel back and it's negative, but this kid still has something. So what's the next step? Generally, the next step is to do whole exome sequencing, right? So now what you're doing is, instead of just looking at those 62 genes that cause that we have know to cause defects in cholestasis, the resulting cholestasis. Now you're going to look at all the different proteins in the body, even proteins that have not historically been associated with a disease, right? There's thousands and thousands of proteins that we know our bodies make, but we don't really know what they do. And we don't know that if they don't work, what happens. And so there, you sometimes get back these hits. So it'll say, here's protein X, when we kind of look at it, it seems like there's something odd about it, right? There may be a defect in this protein X. We don't know what protein X does, and we can't say if this is the reason that you're seeing cholestasis in this child that you're caring for. And so what Dr. Yin has been doing in Cincinnati is trying to figure out if we had that information, okay, well, here's a protein that we don't know what it does. Is there a way that we can somewhat quickly, understand how that may be looking in a different place? And so she has an animal model, which is a fish model. And the reason we like fish is because, they breed 17 times a day, right? So the turnover is quick, right? Mice are quick too, but it still takes, a couple of weeks to make a new mouse. Fish, super easy. So in her little model, she could say, okay, here's gene X. Hey, guess what? Gene X also is present in this fish. If I go in there and I take out gene X, what happens to the fish? Did the fish develop any signs at all of liver disease? And so we kind of call this pipeline of being able to study, take a patient in the clinic, relatively quickly understand where a defect may exist in that patient, and then relatively quickly put that same defect into an animal model to try to study it in a controlled environment. Right? This has always been one of the challenges of PFIC, is that there aren't really any good animal models, and so this kind of bleeds into some of the further talks, right? But because there hasn't, and the fish model is not a perfect model. I think we need to be very clear about that. The zebrafish model is not perfect, but it can suggest that, yeah, you know what, like when you knock gene X out, it looks like the bile duct system in this fish's liver is abnormal, right? I mean, obviously the fish aren't itching, right? We can't assess, pruritus in a fish, but I think you can see that there is a possibility that this gene does have a role in the development of what you're seeing in that child, which is cholestasis. And so that's what Dr. Yan and her group are doing, and it really does kind of it speeds up the discovery process. It doesn't necessarily definitively answer the question. But it can kind of rule things out early on, right? If you take that gene, you knock it out in the fish and nothing happens, well, then it's probably not, that's not the gene. That's not the reason that the child's cholestatic. And so that's, I think, where her work is has been really helpful, is to kind of be a testing ground a little bit for some of these new genes to kind of, say, yep, this one deserves further exploration in a little bit more meaningful way. Does that make sense?
It does make sense. Do you know how many genes have been identified to cause PFIC? Did I ask that correctly?
Well, I mean, so again, the list is growing. I think we have the three classic, the three new ones, and now we have USP53. I tend to, I stop there for now. I think some of these other ones are getting, they are cholestatic, right? But in some of the rare reports, these kids also have a lot of other problems, right? Outside the liver, and a lot of these are being identified in kind of families where there's consanguinity, where kind of the parents are somewhat closely related. And so there's a lot of recessive things coming down the pipe. So that's a tricky one to answer, because probably depending on who you ask, someone may say, I think there's only 3, someone may say 6, someone may say, I think there's 16, because there's rare reports of a lot of these different things. But I am stopping at, I'll stop at 7 for now, but I'm open to more. I just think we need more robust data.
Actually ran into Dr. Thompson this weekend, and he said that he saw, let's see, MYO5B is, if you number it, would be 6. Is that correct?
Yeah.
If you number it. I know
Yeah.
it's an if, but I think he saw it published as PFIC 11. So then, I think the reason I bring that up is because it's in the scientific community, from the way that your knowledge is, advancing forward. I've heard a lot that the numbering system you're starting to do away with the numbering system for good reason. And I think what you just spoke to, and then that is a fairly good example as to why.
Yeah, it becomes hard, right? But it's a lot easier to say PFIC 11 because everyone knows what you're kind of talking about, right? It's a cholestatic baby who probably itches, versus if you say, MYO5B deficiency, a lot of people might not even know what that is off the top of their head. And they definitely wouldn't know, if they'd never heard about it, what the physical manifestations of that are off the top of your head. And so this is where, PFIC, it's going to be hard to fully get PFIC out of the literature because it's so descriptive and it's so good in its description of saying, if you say PFIC, I know exactly what that kid probably looks like.
Mm-hmm.
I know what those labs probably look like. That is helpful information without having to kind of, use a paragraph to get there.
Yeah, it creates confusion for us as a patient community, if you can imagine, which is why I think it's important that we talk about it. We talk about it and we understand the way that you as scientists are thinking of it, and also the way that it makes, that we are understanding it. So it's challenging, and I think it's going to continue to be challenging. So we just have to keep talking about it.
Yep.
Okay, well, in the interest of time, let's move on, because I know that there's a lot of other presentations that were here. This one in particular is something that comes out of Pittsburgh. Is that correct? The modeling PFIC one?
This one's near and dear to my heart because, again, we have a large cohort of PFIC-1 patients here in the Pittsburgh area. And I'm working with this team, but so what this comes back to is essentially what I mentioned before, right, which is that there are no good animal models. And so what we've been doing here in Pittsburgh is this concept of patient-specific stem cells, and what does that mean? Technology is an amazing thing, right? But what we've been able to do for some time, as many of you may know, is if you go back to how a human is made, right? You get a cell from mom, a cell from dad, so you get 2 cells at first, 2 become 4, 4 become 8, 8 to 16, and soon you got a little child. And so, but early on in that process, right, when you're just a group of like a few 100,000 cells, there are signals that work out what goes where, right? So it's like, hey, you guys, you come over here, right? You're gonna go be a skin cell. You guys go over there. You're gonna be bone. You 3 come with me. We're gonna make a liver, right? And that's essentially how a kid gets made. And so what scientists are now able to do is take cells from the skin or from other places. Here in Pittsburgh, we use the skin because it's kind of readily available and easy to use. And they can redirect it, so they can give signals to back up the process and then redirect those cells to become whatever you want. And so we talk about these, we use the term, inducible pluripotent stem cells. But here what you're doing is you're essentially taking, again, in our case, skin cells, backing them up and reprogramming them to become liver-like cells. The advantage here, right, is that you have kept that patient's DNA in the whole process. So the DNA that makes up those liver-like cells are unique to that patient. Now, this is where, I think personally there's going to be an advantage over something like the CRISPR technology that everyone hears about. Because what you do in CRISPR is you're able to kind of, very surgically, insert specific mutations. So for example, if I had a cell line in the lab and I wanted to insert somebody who had, PFIC disease, I could say, yep, what's the defect in that protein? And I could insert that specific defect into the protein of that cell line to study it. But I think going back to the spectrum of PFIC, right, it is clear that it's not just that one defect in that patient that causes all of the manifestations. This is why patients who have the exact same genetic defect will have very different physical manifestations or clinical courses. And so this ability to really make the entire cell the same as the patient, I think enables the opportunity to really personalize how we approach these conditions. And so, here in Pittsburgh, again, we're essentially using these skin cells, reprogramming them into liver-like cells, and then you are able to kind of, in a much more controlled way, study these cells. And we put these cells through the ringer, right? We'll just grow them out and see what they do. We're able to kind of put them into like a single layer and see how well they can secrete bile acids. Some people are using these cells actually to there's an animal model whereby you can actually, essentially kill the rat liver and then immediately inject human cells and repopulate the rat liver with the human cells. So you've got this little kind of human liver inside a rat. There's other kind of, we call them liver on a chip, which is kind of, there's like a 4-cell based platform. So there's just a bunch of different ways that in a lab, one can look to model these disorders by using cells that are unique to patients and that carry that patient's unique DNA signature with them, so that you can try to understand maybe why does one kid present like this and another kid present like that, and or would a medicine work for this kid that might not work for that kid. And you can do this in a shotgun approach, so that you can test 300,000 samples at a time. And this isn't something where you have to like do one at a time. But I caution, right? I mean, every time we talk about stuff like this, parents get all kinds of excited. We get all kinds of excited. It's exciting. But I mean, we are early days in being able to use the information that we're generating from these types of studies in any real meaningful way. I think it is opening doors and it's leading to new areas of discovery, but I definitely don't want to leave the audience with the impression that, in the next couple of years, you'll be able to go to your doctor, take a little skin cell, and 30 days later have a report of medicines that will work for you, and have a personalized treatment plan based on this type of work. But this is kind of what we're trying to do. And then, to be perfectly honest, specifically for PFIC1, we don't know what the FIC1 protein does, right? That's the other kind of frustration. We've never really known what this protein actually does. We think we know some, but we don't know at the end of the day. And so we're also using this model to better focus specifically at that FIC1 protein and try to understand what is its function and why, when it goes bad, you get the phenotype of PFIC.
That's really cool. That's my technical response to it. Is that it's just really cool.
Yeah, and it is really neat. And I think it holds a lot of promise, but I think it is going to require a lot of very careful work to make sure that what we're seeing and what we're doing is actually meaningful to patients. And we are not there yet.
Takes time. Okay. Well, thanks. What I'll do is it's 1:42, so I'll keep moving on, and then we'll come back and do some general questions. And if not, I'll keep asking questions. I think this next topic is something that we've heard of quite often, but it was presented at the conference, and there's been a lot of movement in this area. So maybe a quick update on what Dr. Ricard presented about odevixibat or Bylvay.
Yeah, I mean, I think this is probably the most exciting thing that's happened over the past 10 years with this condition is there's a medicine, right? So we now have a medicine that we can use that really does seem to be impactful for certain patients. Now again, not every patient, and so that's frustrating, and that's something that we need to understand more. But it is essentially a chemical diversion where we're inhibiting the reabsorption of bile acids, and it seems to, as far as we know, really improve at least itch and bile acid concentrations in these patients, or at least in some patients, or quote the responders, right? I think what we still don't know is the long-term effects of this, right? Well, what does it mean 10 years out from now, and is it going to actually change the trajectory of these kids' clinical course? I don't know. Too soon to tell. You can't really do a clinical trial where the endpoint is 10 years away. It's never going to be funded. So, they picked itch and they picked bile acids as their clinical endpoints. They seem to be impactful. And so I think, if what we think we know about the mechanism in which injury occurs in these disorders is true, then in theory, these medicines could really be impactful down the road, but we're going to have to wait and figure it out. I think there's also a lot of excitement about other ways we can use these medicines in these conditions. I think specifically in some of the post-transplant settings. I think even in the diversion setting, right? I mean, just because you've been, it goes both ways, right? That trying to understand, are there patients who will benefit fully or more fully from having both the surgical diversion and taking one of these medicines? And that, we still just don't know that yet. We need to look more into that.
Well, and you're saying one of these medicines. And I think we're all well aware, but I like to kind of talk about these back to back. So this topic was about odevixibat, but then Dr. Looms the next day talked about marilistat, somewhere down here. Yeah. So, and what is the difference? Because when you talk, so what the synopsis you just gave us in relation to odevixibat, that's really IBAT inhibitors in general. Is that correct? So the difference is 2 different drugs. So, and one's in trial and one's been approved by the FDA and the EMA, and it's accessible in some areas of the world. So that's a huge difference. But, would you give, if we went down to the maralixibat talk, would you give the same type of synopsis, really? Both have shown, good data that's working in the patients that, in some patients. And what are the differences in these 2 talks? I guess is my question.
Yeah, none, except for one's further along than the other, to be honest, at the end of the day, right? So as many of you I'm sure may know, maralixibat is approved, at least in the United States, for Alagilles, right? One of those other rare cholestatic liver conditions that can share a lot of similarities with the PFIC as it relates to cholestasis niche. And this, I wasn't in the room, obviously, at all when in these, but, it appears to me that this is just, the drug company initially deciding, you can't do it all at once, right? You got to kind of, put your efforts behind, you got to start somewhere. And it seemed that the maralixibat group focused initially on getting approval, at least in the US, for Alagilles. The odevixibat group, went for the PFIC disorders. They are both now switching, right, and trying to get approvals for both of those things. Also, there's, a dozen other IBATs in the pipeline, that are being studied in other various conditions like fatty liver disease, like primary sclerosing cholangitis, like primary biliary cirrhosis, which is really a disease of adults. Any disease, liver disease, biliary atresia, so there are going to be many more of these drugs. I think the indications for them will expand. I think that's only good news for the PFIC community, because although in theory, they have a similar mechanism of action, I think clinically, in other instances, let's take, for example, proton pump inhibitors for acid suppression, right? Whether it's Nexium or Protonix or, whatever, I think clinically, although these are the same class of medications, some patients seem to do better with one versus another. And so although all IBAT inhibitors, right, if it has IBAT at the end, it is an ileal bile acid transport inhibitor. That's what, all of these will be blah, 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 IBAT, right? Because that kind of helps us clarify the class of drug, having options as it relates to some of these medicines is going to be beneficial. So I think that I would likely give the same information for this as I did for that last talk. It just happens to be that, at least in the United States, the maralixibat drug is now being focused on the PFICs as opposed to the odevixibat, which has obviously been approved. If I was giving this talk to the Gala Alliance, I would switch that.
Fair enough. Thanks. Okay, well, I'm going to scroll back up. I'm going to skip over the roundtables for now, so that we can do a talk breakdown. And if we have a chance, we can summarize the roundtables. And I'm going to encourage anybody who has a question to maybe enter it into the chat, so I know to ask it, because Dr. Squires does have to leave promptly in 10 minutes. But let's go on. So that takes us through day one. I know the community our community members gave some really powerful talks and some shared their stories as well. And we got to hear a wonderful introduction from a patient who shared her experience with it. And then this community keynote, these family stories from around the world, was really powerful in terms of hearing from others in other areas of the world. Our experiences are very similar. And we unite together, really, in PFIC, and we're one big community. And I think they did a wonderful job sharing. Let's see. So day two, the scientific talks. We heard about ileal bypass, which I was excited about because there's been so much talk about the IBAT inhibitors, but ileal bypass is just as much a part of our realm. Has been for a lot longer. So Dr. Czapkowski gave a really a great talk about ileal bypass and a case study as well, which was very powerful. So maybe speak to that one a little bit.
Yeah, I mean, this is, ileal bypass. I think I was excited about this talk as well, because this just comes down to institutional experience, right? These diversion surgeries, whether it's partial external biliary diversion, whether it's partial internal biliary diversion, whether it's ileal bypass, a lot of this just comes down to, where you land and what the surgical team and the hepatology team, the liver team at that place is doing. There has never been any comparative effectiveness trials that have looked at these surgical options and in any meaningful way demonstrated benefits of one over the other. And I think you could pick off advantages and disadvantages of each. And I think it is fair to ask as a patient, right, which should I go for, right? If you're telling me there's three options, which should I go for? What I would say if you came here is we do partial external biliary diversion. That's been our practice. If you came and said we demand an ileal exclusion, would we do it? I don't know. I'd have to talk with our surgeons, right? But we haven't done too many of them, and I think that our experience with external diversion is so good that we try to talk you out. Not because I'm right, right, but it's just because it's what we do. And so I think that as a patient, and again, I know that probably is more frustrating to hear than anything else, but I think that all of these surgeries do seem to make a difference, and at the end of the day, hopefully the surgeries go away as the medicines become more effective, because they're all doing the same thing. But I think keeping all of these and continuing to have these small reports out there, right? I mean, a lot of people will look at even the data he presented and be like, this is a one case series of a handful of cases. You can't draw anything meaningful from that, and you got to be careful, because you really can't, right? We can't take one report of one center's experience, whether it's mine or anybody else's, and live by that law. But I think it just these things continue to add to the literature. So I think all information is good information, but I think what they're doing in Poland, I think is fantastic. It still wasn't clear to me after his talk, did they favor this versus one of the other experiences, right? I kind of wanted to push him on that, but I didn't, because I think if you are at a center that does multiple, how do you decide? And I don't know how one decides, because there's not enough data to tell you that one's better than the other.
Yeah. No, that makes a lot of sense. Well, maybe we'll have another opportunity at the next conference to ask him. There's a comment in the chat. Can you touch on the talk someone gave where they mentioned having to take anti-rejection drugs after gene therapy? And I think that was Dr. Asai's talk.
Yeah.
So maybe we can skip to that one and then go back to Dr. Menon's talk.
Yeah. I mean, again, so gene therapy is another one of these buzzwords and promises that the field has yet to fully live up to, right? And there's different ways in which people provide gene therapy, but what Aki was trying to do here was kind of connect the fact that in gene therapy, right, and this was more in an animal model, right, then this hasn't been done in humans yet, but in gene therapy, where you deliver a new gene to a patient, you generally do that by giving a virus that's been genetically modified to insert the new gene into the patient, right? That's how gene therapy typically works. Now, the concern for gene therapy, specifically for BSEP, is that we know from our transplant experience that patients who get transplanted for BSEP can develop antibodies specific to the BSEP protein, which essentially nullifies the transplant, right? Because it attacks that singular BSEP protein, destroys it, your immune system does, and so you can revert back to essentially having a liver without a BSEP protein. Now, we have developed surveillance, and now that we know that happens, we watch for it, and there are interventions that we can do to prevent that or treat that. Again, the numbers are so small that it's hard to say that our treatment is perfect and that it is, will always work. But I think knowledge has allowed us to at least have a plan for if we see that occurring. But as we talk about gene therapy, I think what Aki wanted to try to understand is if we gave gene therapy instead of a blunt whole liver transplant, if down the line we're able to do just gene therapy in a patient, would we have to watch for that same recurrence? And so that's, and if you do, and so that's what he was showing is that, if you do gene therapy in someone who has BSEP deficiency, it may be that they will still need to take quote unquote, anti-rejection medicine or autoimmune modification medicines in order to prevent the body from making antibodies against that BSEP protein that's been delivered. So I know that's still a little bit of a more technical answer than I like to give, but did that answer the question?
I think so. I think it was a pretty good summary. I'll open it up to anyone in the chat who has a further question to clarify, because I know it's kind of a challenging topic.
Yeah, I mean, I guess, while maybe things are coming in, for example, if we were to have a gene therapy for FIC1, right? It has not been our experience in the transplant setting that you can develop antibodies to the FIC1 protein after your liver transplant. So we would not think that if you got gene therapy for FIC1, you would need to be worried that your body would attack that single protein if it got into the liver. But in BSEP, you do. And so I think that was his talk, was that, not so much that this is something that's happening now, but if gene therapy ever were to become something that could be offered to a patient with PFIC, at least for the BSEP disorders, the animal models would suggest we also still need to be worried about this recurrence, even though it's gene therapy and not a whole organ transplant.
Okay, interesting. Yeah, good to know. This one obviously hits home to me because my daughter is one of these patients who has reoccurrence of disease post transplant, and it's another one of those if we knew now what we knew then type thing. Back when my daughter was getting transplanted, this was rarely talked about. I think it was one or two other patients had this experience, and it was a conversation between me and Cedar's doctor that kind of led to this discovery, and it was manageable, and it ended up being, it's managed. I know it's not always managed, but it's talked about more, and it seems to be understood a bit better, and it seems as though there's interventions that can help keep this from causing too much harm at times. Just one of those advancement comments. We really only have 2 minutes. So maybe a very quick touch base on Dr. Meenan's talk for liver transplantation and TJP2 deficiency. And then we'll wrap up, and I can take questions via email and send them to Dr. Squires and vet them that way if we didn't get to anything.
I think another great talk of, a very small experience with a very rare disorder. Any number of these disorders, we feel like patients can progress and may need liver transplants. The reports, again, particularly in the 4, 5, and 6s up to 11, wherever you want to count, right, of the role of liver transplant for these disorders, the more reports we get, the more information we're going to have. And so again, this comes back to what I said before about these single center experiences where the numbers are small, still do add to the literature, because right now, if I had a kid who came in with TJP2 and really significant severe liver disease, and the family said, well, if we do a liver transplant, how is that, is that indicated? What are the outcomes? I would say there's really very little data. And so work like this just adds to my ability to have those conversations with the patients about what we would expect. Yes, it's been done before. These are the outcomes. This is what, kind of has happened to these patients. And so, providing these experiences to the community to allow us to have more informed conversations is what's going to be important. You could take out TJP2 and put in MYO5B, put in FXR, put in, whatever the next PFIC is. I think it's pretty established what post-transplant looks like in FIC1 and in BSEP, because we've done a lot of them, and there's been a lot of reports about what happens to these kids after transplant. There just isn't a lot of reports about some of the rarer PFICs, and that's that's a relative term, right? But some of the less known PFICs, there just isn't that many reports in the literature about what transplant outcomes may look like. And so I think this was just added to that clinical experience, which I think is still, it's going to be helpful to help frame conversations.
Okay. Well, thanks. Yeah, I mean, it's just a testament to the more you see, the more you learn, the more you know. So thank you and all your colleagues who presented at the conference, your global colleagues, I'll say, and for all of the work that you continue to do to try to understand the disease that affects our patient community. So yeah, with that, I know that you have to run. So thank you for spending your lunch break with us, I believe. If anyone has any questions that they want to send to Dr. Squires, please feel free. You can send them to me and I can pass them on, get you in touch. And if we want to invite Dr. Squires back, we can do that if we want to hear more. So thanks, Dr. Squires. I really appreciate it. We really appreciate it.
Hey guys, take care.
Bye-bye.
See ya.
Thank you.